Let me ask you something. What if your health depends less on calories and more on electrons? Not metaphorically, literally. Your body runs on controlled electron flow. Mitochondria pass electrons down a chain to make ATP, also known as one of our sources of energy. Cells signal using redox reaction, that means reduction and oxidation. Even your blood depends on electrical charge. And this is where polyphenols become far more interesting than just antioxidants. Polyphenols are electron donors. At their core, polyphenols are structured electron donors. They contain aromatic rings with hydroxyl groups. At their core, polyphenols are structured electron donors. They contain aromatic rings with hydroxyl groups. Those OH bonds can give up electrons. When a reactive oxygen species is looking to steal an electron, a good polyphenol says, "Take mine." That stabilizes the reactive molecule. And because of the ring structure, the polyphenol can spread out the charge and remain relatively stable itself. That is elegant redox chemistry. Oxidative stress is not some abstract concept. It is electron imbalance. Too many unstable molecules trying to grab electrons. Too few safe donors. Polyphenols help restore that balance. But not all polyphenols are equal. A 2025 study examined how structural changes in polyphenols altered their ability to reduce toxic chromium-6 to the safer chromium-3. What determined performance? How good an electron donor they were. Polyphenols with stronger electron-donating groups reduce chromium-6 faster. Why? Because they had lower ionization energy. They had weaker OH bond strength. In simple language, they gave up electrons more easily. Structure dictated generosity. Small changes in chemistry meant big changes in electron flow. That is redox biology in action. And so let's talk about zeta potential for a minute and why charge matters. Here we're going to talk about something most people never connect to nutrition, zeta potential. Zeta potential measures surface electrical charge. If particles carry strong surface charge, they repel each other. That keeps them dispersed instead of clumping. In the chromium study, polyphenols with stronger absolute zeta potential values stayed better dispersed and interacted more effectively. More dispersion means more surface contact. More contact means better redox interaction. But here's where this gets even more interesting. Your red blood cells also have a zeta potential. They carry a negative surface charge. That negative charge causes them to repel each other, which prevents clumping. If red blood cells lose that negative charge, they aggregate. Blood becomes more viscous, microcirculation suffers. So surface charge influences blood flow. Blood flow influences oxygen delivery, and oxygen delivery influences mitochondrial ATP production, energy production. See the chain? Redox state, oxidative stress, membrane integrity, and surface charge all interact. Polyphenols, by influencing redox chemistry and potentially interacting with membranes and proteins, operate within this electrically sensitive environment. They are not just mopping up radicals, they participate in a charged biological system. So redox biology can be thought of as organized electricity in the body. Redox is controlled electron traffic. Healthy cells maintain balance between oxidation and reduction. Too much oxidation damages membranes, proteins, and DNA. Too much reduction is not good either. The body thrives in controlled tension. And polyphenols contribute by donating electrons when needed, stabilizing radical intermediates, influencing surface chemistry, participating in metal redox cycling. And they are part of a network of electrical buffering. And when that buffering works well, mitochondria function efficiently. Inflammatory signaling is regulated. Cell membranes remain stable and microcirculation stays smooth. So the bigger shift here is that food is not just fuel, it is electrical potential packaged in molecular form. Polyphenols are structured electron reservoirs. Their power depends on the position of hydroxyl groups, the strength of electron donating substituents, and ease of electron transfer. Surface charge behavior reflected in zeta potential. Tiny molecular tweaks can dramatically change redox performance. That is precision biochemistry. So the next time someone says antioxidants are good for you, ask a better question. How well do they donate electrons? How stable are they after donation? And how do they behave in a charged biological environment? Because health at its deepest level is organized electron flow. If this changed how you see food and microbes, share it with someone who needs a better explanation than antioxidants are good. And if you want to rebuild gut stability by restoring energy production and redox balance at the root, sign up for my newsletter by watching the masterclass link below. I share regular content on underlying causes of gut inflammation and what can help.